Hello everyone. Today we'll have the revision on carbohydrate metabolism. In this session, we'll be focusing on the questions on glycolysis, significance of 2,3-BPG shunt, important product of Rapoport-Luebering cycle in RBC, and the role of fluoride bottle for the blood collection, pyruvate, and pH complex. So let us discuss one by one these questions and how to write these answers. Already we have been learning detail about all these sessions in the first few months while learning the chemistry and metabolism of carbohydrate in detail. So here we are not going to teach you again in detail, but we will give an idea how to write the answer for the examination. Now the first question is regarding glycolysis. Describe by a flow diagram the reactions of glycolysis, giving names of enzymes involved. Explain its regulation and add a note on the energetics in aerobic and anaerobic conditions. So this question was asked in KUHS in previous year as a 10-mark question. But yeah, for the additional batch it can be continued as the same 10-mark pattern. For the regular batch it will be as for a 15-mark question. So whatever it is, your answer should have these points in your answer, that is definition, site where it is occurs, key enzymes, pathway, in between I forgot to write, that is the significance of pathway, then regulation and energetics. So you should have a definite definition for the glycolysis. We know that glycolysis is the oxidative pathway, oxidation of glucose into finally pyruvate. The splitting of 6 carbon glucose into 2 molecule of 3 carbon pyruvate aerobically and anaerobically into lactate. That is should be your definition. And regarding the site, where does it occur? It is occurs almost all the cells and it occurs inside the cytoplasm. And you can have the significance of glycolytic pathway. Specifically, you can write that. So, what are the significance? Is only pathway that is taking place in almost all the cells in our body, and it occurs mainly in the cytoplasm. And remember, it is the only source of energy in RBC. And in vigorous exercises, when the muscle tissue lacks oxygen, that is during anaerobic condition, anaerobic glycolysis, which forms a major source of energy for the muscles. And it is a preliminary step before the complete oxidation. And it provides the carbon skeletons for the synthesis of non-essential amino acids. And also it provides the carbon skeleton for the glycerol part of fat. And most of the glycolytic pathways are reversible, and those steps can be participate for the gluconeogenesis. And it is mandatory to maintain a normal blood sugar level. So you should know the normal blood glucose level, that is 70 to 110 milligram per deciliter. So after heavy carbohydrate meal, it usually goes very high, and in a normal person which will be below 150 milligram per deciliter. So this should be your introductory part of your essay. Then you can go and directly draw the pathway. So pathway you have to draw perfectly with enzymes and coenzyme. Your enzyme should be proper, your coenzyme should be proper. Without enzyme and coenzymes, your essay will be incomplete. So let me brief the pathway. So here a 6 carbon glucose molecule will be 6 carbon glucose will be breaking down into 3 carbon pyruvate, 2 molecule of 3 carbon pyruvate. So first glucose undergoing phosphorylation with the help of hexokinase or glucokinase enzyme with the hydrolysis of ATP to ADP, and your intermediate is glucose 6-phosphate, and hexokinase and glucokinase which requires magnesium as the cofactor. Then it undergoes an isomerization reaction with phosphohexose isomerase to form fructose D phosphate. Again, a second phosphorylation occurs to fructose 6-phosphate by PFK enzyme, that is phosphofructokinase, to form fructose 1,6-bisphosphate. These three, step 1, 2, 3 is known as the preliminary or preparatory step of glycolysis. Now this step, the coming step which explains the breakdown step. Here a 6-carbon fructose 1,6-phosphate will be breaking down into 2 molecules of 3-carbon molecule, that is one your dihydroxyacetone phosphate and the second glyceraldehyde 3-phosphate. It is done with aldolase enzyme. Here the dihydroxyacetone phosphate can be isomerized to glyceraldehyde 3-phosphate. So in the remaining cycle or remaining steps, 2 molecules of glyceraldehyde will be undergoing the remaining step. So 2 molecules of glyceraldehyde undergoing an oxidative phosphorylation with glyceraldehyde 3-phosphate dehydrogenase, where NAD accepts the hydrogen to form 1,3-bisphosphoglycerate. And this NADH can enter into ETC and can get the ATP. It is written 3 ATP here, but according to the new concept it is 2.5 ATP. This occurs during aerobic glycolysis. Now this 1,3-bisphosphoglycerate undergoing a substrate level phosphorylation. Here what will happen? Phosphate group will be, one phosphate group will be eliminated to form 3-phosphoglycerate, and the phosphate group is accepted by ADP to form ATP. So that is why this is a substrate level phosphorylation. The previous one was a oxidative level phosphorylation. Now the 3-phosphoglycerate undergoing a mutase reaction to form 2-phosphoglycerate. Further, enolase enzyme helps the removal of a molecule of water from 2-phosphoglycerate to form phosphoenol pyruvate. This phosphoenol pyruvate again undergoing a substrate level phosphorylation to remove the phosphate group from this structure by pyruvate kinase enzyme to form finally pyruvate. So this is the end product during aerobic glycolysis. During anaerobic glycolysis, this pyruvate undergoing reduction by lactate dehydrogenase, where the NADH here utilized will be from the step glyceraldehyde 3-phosphate dehydrogenase step, that will not enter into ETC, instead of that which will be utilized here and which will be leading to the product as lactate. So here in this step you have to remember that the step 1, step 3 and step 9, that is hexokinase, phosphofructokinase and pyruvate kinase. These three are the key enzymes of glycolysis. These three are the irreversible steps of glycolysis. This cannot be reversed, whereas the remaining steps are reversed, which can participate in gluconeogenesis. So these three steps are the regulatory enzyme which helps to regulate the glycolysis. So this is overall about the glycolytic outline pathway. Remember, once you write this properly, no need to write the steps again and again. Now regarding the energetics. This explains the, this is oxygen deficiency, that is anaerobic glycolysis. Anaerobic glycolysis, you have to remember that the 1,3-bisphosphoglycerate, that step which convert to 3-phosphoglycerate, does not, which usually synthesize NADH, usually which will be utilizing for the, which will be entering for the ETC. Now here that NADH will be utilized here for the reduction of pyruvate to lactate. So 2 molecule of NADH will get usually 5 ATP. That does not occur in anaerobic condition. So the remaining step, that is step 1 and step 3, usually which utilizes the ATP. So we have to subtract those 2 ATP. Then 2 substrate level phosphorylation, that is 1,3-bisphosphoglycerate kinase and pyruvate kinase enzyme, which leads to the production of 2 molecule of ATP. So 2 plus 2, 4 ATP, 4 minus 2, total 2 ATP only during anaerobic glycolysis. Whereas in aerobic glycolysis, you can see all those things. Step 1 and 3 , 2, that is 1 ATP utilized. Step 5, 1 ATP utilized. Step 6, 2 ATP utilized. Step 7, 1 ATP utilized. Step 8, 2 ATP utilized. Step 9, 1 ATP utilized. Step 10, 1 ATP utilized. Step 11, 1 ATP utilized. Step 12, 1 ATP utilized. Step 13, 1 ATP utilized. Step 14, 1 ATP utilized. Step 15, 1 ATP utilized. Step 16, 1 ATP utilized. Step 17, 1 ATP utilized. Step 18, 1 ATP utilized. Step 19, 1 ATP utilized. Step 20, 1 ATP utilized. Step 21, 1 ATP utilized. Step 22, 1 ATP utilized. Step 23, 1 ATP utilized. Step 24, 1 ATP utilized. Step 25, 1 ATP utilized. Step 26, 1 that is glyceraldehyde 3-phosphate dehydrogenase enzyme. We have got 5 ATP through NADH, which goes to the ETZ and get 5 ATP. Then step 6 and 9, again 2 substrate level phosphorylation. So total 5 plus 2 plus 2, 7, 5 plus 2 plus 2, that is 9 ATP overall produced. We have to subtract the initial one which we utilized for the first and third step. So total 9 minus 2, 7 ATP generated in the anaerobic glycolysis. Now regarding the regulation of glycolysis. So the hormonal regulation, which is mainly done with the insulin which furthers glycolysis, and glucagon has opposite effect. Glucagon and glucocorticoids which inhibit glycolysis. So insulin usually which furthers glycolysis by activating the key glycolytic enzymes as glucokinase, phosphoruptockinase, pyruvate kinase, etc. And PFK, that is phosphoruptockinase, which is the rate limiting enzyme for the glycolytic pathway, which usually allosterically regulated by factors like the inhibitors are ATP and citrate, activators are AMP and fructose 6-phosphate. So here again same thing, where is enzyme and what are the factors which are activating and inhibiting. You have to go through all things. Then this figure which again explains the hormonal regulation. Insulin stimulate the key glycolytic enzyme. Glucagon inhibit key glycolytic enzymes. The key enzymes are glucokinase, phosphofructokinase, and pyruvate. Okay, let's go to the second question for the discussion. Significance of 2,3-BPG. 2,3-BPG is also known as Rapoport-Luebering cycle of erythrocytes. It is a shunt pathway. So usually in glycolysis, 1,3-bisphosphoglycerate will be undergoing substrate level phosphorylation to form 3-phosphoglycerate. Here in RBC, 1,3-bisphosphoglycerate which undergoing a shunt pathway by bisphosphoglycerate mutates to form 2,3-bisphosphoglycerate. This 2,3-bisphosphoglycerate has some significance in RBC that I will explain. This 2,3-bisphosphoglycerate, by removing phosphate by 2,3-bisphosphoglycerate phosphatase, it again can be converted into 3-phosphoglycerate. Now in RBC, what is the role of this 2,3-bisphosphoglycerate? We have to see. So significance is that when 2,3-BPG combines with the hemoglobin, it reduces the affinity towards the oxygen. So what is the role? It helps to unload the oxygen to the tissues. So in presence of 2,3-BPG, oxyhemoglobin will unload oxygen more easily in the tissues. So under hypoxic condition, 2,3-BPG concentration in the RBC increases and furthers the release of oxygen to the tissues even when the partial pressure of oxygen is low. And the compensatory increase in 2,3-BPG in high altitudes favors oxygen dissociation, and it is increased in fetal circulation. Remember, there is no ATPs generated in 2,3-BPG shunt or Rapoport-Luebering shunt. So even this question can be asked, Rapoport-Luebering shunt. In that also you have to write the same thing, draw the pathway and write the significance of this pathway. Rapoport-Luebering shunt or 2,3-BPG shunt is one and same. Another question asked was important product of Rapoport-Luebering cycle in RBC. Answer is 2,3-bisphosphoglycerate. Now the next question from this session is that explain why glucose is collected in fluoride bottle for glucose estimation. So if you know the glycolytic pathway properly, in that I have said fluoride will be inhibiting the enzyme enolase. Enolase enzyme actually requires magnesium as a cofactor. So this in fluoride which causes the displacement of magnesium from the reaction, so the enolase activity does not occur properly, so that what will happen, the glycolysis will be getting inhibited. So it helps to prevent the glycolysis in blood prior to the estimation of blood glucose. So Thereby we can getting an accurate blood sugar value. So if you are not adding fluoride to the tube before blood collection, what will happen? Glycolysis continues by the living tissues or living cells which present in the blood will utilizes the glucose through the glycolysis, and will get a low value compared to the original value, so that we have to add fluoride to inhibit the glycolysis. Now next question is fate of pyruvate. So under aerobic condition, pyruvate is converted to acetyl-CoA, which enters the TCA cycle to be oxidized to carbon dioxide. We know that the final fate of pyruvate is that pyruvate has to undergo TCA cycle for the complete oxidation to produce carbon dioxide and water, and concomitant production of energy. Glycolysis is taking place in cytoplasm, so the pyruvate is also generated in cytoplasm, but it has to transport it into mitochondria by pyruvate transport. Once it reaches there, it has multiple fate. This pyruvate can undergoing oxidative decarboxylation by PDH, that is pyruvate dehydrogenase enzyme complex, to form acetyl-CoA. This acetyl-CoA can be entered into TCA cycle, or it can be even entered into, therefore the fatty acid synthesis depends on the situation. That is one fate. Or pyruvate during anaerobic condition, what will happen? It is reduced to lactic acid or lactate by lactate dehydrogenase enzyme. Even pyruvate can undergoing transamination by alanine transaminase enzyme to form alanine. So this is again the non-essential amino acid can be synthesized by using carbon skeleton. Or the pyruvate can undergoing carboxylation by pyruvate carboxylase enzyme to form oxaloacetate, and which can be getting participate into the gluconeogenesis through the other steps. This oxaloacetate has multiple fate. Oxaloacetate even can be undergoing malate dehydrogenase enzyme reaction to form malate, or it can undergoing transamination to form aspartic acid, or oxaloacetate can be going to the gluconeogenetic pathway through phosphoenol pyruvate carboxy kinase enzyme reaction. So this is all about the fate of pyruvate. So this figure telling that glucose to fatty acid is possible, but fat to glucose is not possible because PDH reaction is completely irreversible. The next question was PDH, pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex is located in the mitochondrial matrix. It is a multimolecular aggregate consisting of multiple enzymes, 3 enzymes mainly, and 5 coenzymes. This PDH is responsible for the conversion of pyruvate to acetyl coenzyme. The reaction is oxidative decarboxylation. So the enzyme part of PDH complex is that pyruvate decarboxylase or pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase enzyme. And the coenzymes are thymine pyrophosphate, lipomide, coenzyme A, FAD, and NAD. And remember, pyruvate to acetyl coenzyme reaction is completely irreversible reaction. So, what is the purpose of being this multi-enzyme complex? So, aggregation of various enzymes in a complex which usually increase the efficiency of the transformation of the reaction. Why? Because the intermediate products are not released in the medium easily, so that which can be channeled to the succeeding enzymes of the pathway. So a metabolic intermediate is always available for the next reaction immediately after its protection. So that is the advantage of being a multi-enzyme complex that we have already learned with fatty acid synthase complex also. Even though the reaction looks very simple, like pyruvate undergoing oxidative decarboxylation by PDH complex to form acetyl-CoA, but it is a complex reaction. So let me try to explain. First, the pyruvate and thymine pyrophosphate combine to form a condensation product. Later, that is done with pyruvate dehydrogenase enzyme. Then, pyruvate decarboxylase enzyme, which help for the release of a carbon dioxide from this condensation product, and we are getting an intermediate known as hydroxyethyl thymine pyrophosphate. This is attached to the reactive carbon of the thymine pyrophosphate, that is hydroxyethyl group will be attached to. Then next reaction is transfer of acetyl group from the hydroxyethyl intermediate to the lipoic acid. The acetyl group will be transferred to the lipoic acid to form acetyl lipoic acid. And this is catalyzed by dihydrolipoyl transacetylase enzyme. Next, the step 4 is that transfer of an acetyl group from the acetyl lipoic acid to the coenzyme A molecule. The acetyl coenzyme A then enters the TCA cycle. So the acetyl coenzyme A will be eliminated and this enters the TCA cycle. The remaining product is reduced lipoic acid. This step regenerates the oxidized lipoic acid so that we can again go and continue with the reaction. So this is all about the PDH complex reaction. Now the regulation of PDH is done with end product itself, that is acetyl coenzyme itself will be inhibiting the PDH complex enzyme and NADH also. Covalent modification of PDH enzyme occurs by the end product. And the clinical aspects is in thiamine deficiency, PDH activity will be decreased and the pyruvate is converted into lactate. It leads to lactic acidosis. Second one, inherited deficiency of the glycolytic enzyme like pyruvate kinase and aldehydes. So this is all about the question discussion on glycolysis 2,3-BPG shunt. Then we have seen the role of fluoride for the blood collection. Then we have seen the ferropyruvate and PDH complex. Thank you all.